Friends, welcome to Biology Made Easy. Today we are starting a new chapter, that is the cell, the unit of life. And in this chapter, the first video that we will discuss today is prokaryotic cell. Prokaryotic cell. Now from this prokaryotic, you can guess. Now pro means primitive and karyon means nucleus. That means the cell in which the nucleus is primitive is known as prokaryotic cell. So what is the meaning of this primitive nucleus? Usually the nucleus is surrounded by a nuclear membrane in case of higher organisms. But in case of primitive organisms, the nucleus is not surrounded by a nuclear membrane. They are not associated by histone proteins. They are circular. So they are known as prokaryotic cell. That means primitive nucleus. And what are the examples coming under this prokaryotic cell? Usually the different examples are eubacteria, cyanobacteria, mycoplasma, PPLO. These are the different examples of a prokaryotic cell. So when we will discuss the structure of a prokaryotic cell, we have to discuss all the aspects, that is what is the size, shape and structure. So first we will discuss the size. Usually most of the prokaryotic cells, they range from 1 to 2, that is a micrometer, while PPLO, which is the smallest one, it is only 1 micrometer, while most of the prokaryotic cells, they range from 1 to 2 micrometer. And coming to the shape, they may be round when it is, or spherical when it is known as coccus, or they may be elongated when it is known as bacillus, or they may be spiral shaped when they are known as that is a spirillum, and sometimes they may be the punctuation comma, so that is known as vibrio or comma shaped. These are the different types of the shapes present in bacteria. These are the common types. Then next one we will discuss the structure. The outermost part of the prokaryotic cell is known as cell envelope. And this cell envelope consists of three components. The outermost one is known as glycocalyx. Then inner to it is known as cell wall, and then cell membrane. These three together is known as cell envelope. Combined it is known as cell envelope. So what is the glycocalyx? Now let us draw the diagram from which everything will be clear. Now this is the cell wall. Then out of the cell wall there is this glycocalyx, and this cell wall is we'll discuss. This is the cell wall, and out of the cell wall there is a glycocalyx. So this is the glycocalyx. Now this glycocalyx, it may be, usually the glycocalyx is made up of that is polysaccharides. It is made up of polysaccharides, but these polysaccharides are non-cellulosic. That means cellulosic materials are not there, but they are made of polysaccharides with or without proteins. With or without proteins. And if it is very loosely arranged, this is the glycocalyx. If it is loosely arranged, then it is known as slime layer. But if it is very rigid and thick, it is known as capsule. Suppose here it is it is very thick. Suppose it is thick, then it is known as capsule. And if it is thin, then it is known as slime layer. Slime layer. So if the capsule is present, that means it is rigid, then it is known as smooth. It is smooth, or we can tell that the bacteria is S type. S type. And if this capsule is absent, then we will call it as R type. R type. That means the bacteria may be smooth type or rough type, depending upon whether capsule is present or absent. And it is usually made up of polysaccharides with or without protein. Now, what is this function of this glycocalyx? Now, usually one of the important function of this glycocalyx is acts as antigenic or virulence. That means only if it is smooth type, only if its capsule is there, then it can infect others. So that is one of the problems. Then that is prevention from virus as well as from macrophages. Then another important function is this makes the bacteria sticky, so that it can be attached to any surface, and also it protects it or prevents desiccation. So these are some of the important properties of this sticky nature is very important for its own survival, and it is antigenic so that it can infect others. So this is the structure and the types, that may be slime layer and capsule, depending upon S type and R type, and these are the functions. Then below this glycocalyx, this layer is known as cell wall. This is the cell wall. Usually, this cell wall is made up of the cell wall is usually made up of that is we can tell mucopeptides, or we can tell that is maybe peptoglycan. So pepto means protein, glycan means carbohydrate. Here also muco means carbohydrate and peptide when protein. That means anything we can tell that is mucopeptide or peptidoglycan. So in the protein part, there are 4 amino acids which are even the form of a short peptide bond. And the carbohydrate part, there are 2 things, that is NAG and NAM. They are present alternately. This is N-acetyl glucosamine, this is N-acetyl muramic acid. They are present alternately. So this is the cell wall that is present. Now this, if we will use that is stain, like that of the Christian Gram stain, some of the cell walls, in some bacteria the cell wall will turn purple or blue, and in others it will be colorless. If that purple color is retained, then the bacteria is known as gram-positive bacteria, and if it becomes colorless, it is known as gram-negative bacteria. So, how it will know it? Now, if we will give a crystal violet, that is alkaline crystal violet to the bacteria, and then treat it with 0.5% of iodine, then wash with ethyl alcohol or acetone, and finally wash with water. Then 2 things will happen. Some will retain color, some will not retain color. Those which will retain purple color, because after removing this crystalline violet, all will be purple. But after treating with this ethyl alcohol and washing with water, Those which are negative, they will be colorless. So those which will retain purple color, they are known as gram positive, and those which are colorless, they are known as gram negative. That means depending upon this is known as gram scale according to the name of Christian Gram. So the bacteria are again of two types, they may be gram positive or gram negative. There are so many differences between gram positive and gram negative bacteria, but that is not our point of discussion at present. When we will discuss the Monera, we will discuss all the details. So depending upon this glycocalyx, they may be smooth or rough type. Depending upon the cell wall, they may be gram positive, gram negative, depending upon whether they are retaining the color or not. Now the third one is the cell membrane. Now within the cell wall, there is a cell membrane, and the cell membrane has sometimes certain infoldings are there. Here and there some infoldings are there. This is known as cell membrane. This is cell membrane. And the cell membrane is similar in structure like that of the eukaryotes. It is made up of only that lipoproteins, just like that of the eukaryotic cell made up of lipoproteins. The only difference is that in case of eukaryotic cell membranes, cholesterol or sterols are present to provide stability, but in case of prokaryotic cell, sterols are absent and only they are replaced by hopanoids. Hopanoids, that means in the cell membrane, instead of sterols, hopanoids are present, which maintain the stability of the cell membrane. Otherwise, it is made up of lipoprotein structure. So these three together is known as the cell envelope. Then the next thing is come to see this diagram where most of the things will be very clear. Now see, the cell membrane has some foldings like this. These are known as mesosomes. This is known as mesosome. And this mesosome is just the substitute of a mitochondria that is found in case of the eukaryotic cells. Because this mesosome is important because it is it contains the respiratory enzymes. Or you can tell it carries out respiration. This carries out also secretion. The third one, it helps in that is cell division, and also it helps in DNA replication. DNA replication. And since it is folded several times, it increases the surface area so that there can be maximum number of physiological activities. Increase in surface area. These are the functions of the mesosomes. Now, to this mesosome, when we have written DNA replication, it helps in DNA replication. That means the DNA, which is present in case of prokaryotes, that is attached to this. Then only it will help in replication. Otherwise, how it will help? So, that regarding this we will discuss later on. Now this cell membrane has folding, this is known as mesosome. In addition to this mesosome, sometimes from the cell membrane, there are some other foldings like this, some other foldings. Now these are known as chromatophores. Chromatophores, and these chromatophores, they contain the photosynthetic pigments in case of the cyanobacteria and purple and green bacteria which carries out photosynthesis, but they are not present in other cases. So that is known as chromatophore. Then, like the eukaryotic cells, in eukaryotic cells so many membrane-bound organelles are there, plastids, mitochondria, endoplasmic reticulum, Golgi body, all those there. But all the membrane-bound organelles are completely absent here in the cytoplasm. But here we will get some organelles which are membraneless, they are the ribosomes.